All right, let's get going. Thank you very much for setting everything up. And it's really nice to be back in person. The last one we had, like Kavita was saying earlier, was online, and it was less than fun. I mean, we did what we could. So today's talk right now is about 45 minutes or so, and it's going to be a lot of very basic material. This is just to make sure that everybody's on the same page. This is a time if you have any confusion about anything that's basic, don't worry about it, just ask. It's better to get that clarified now. Lectures will get a little bit more involved and intense over time. And so it's important that everybody is on the same page in the first day or two, okay? We've had an unfortunate cancellation. So Himani Sachdeva, who was supposed to be teaching in the school, is very ill. And so she is unable to teach. So there'll be some schedule rearrangements. Please bear with us. We're going to try and make sure that you still learn what is important to learn. There will be some disruptions. Okay, good. So today's discussion is largely going to be on variation and what generates variation and what are the forces that act on this variation to then sort of lead to changes over time, which we call evolution in natural populations and so on. So just some key concepts, again, so that everybody's on the same page. Please stop me at any point if something is not clear or if you want me to discuss something more. How many people are familiar with this, what I have up here at the moment? These are sort of these main evolutionary forces that we think of. Once you have variation in a population, what does evolution involve? We typically define evolution as changes in allele frequencies over time, right? And these are the forces that can change eventually allele frequencies over time. So here, migration, mutation, and recombination are three important ways in which new variation can be introduced in populations, right? Why migration? Because any individuals that are coming into a population from a different population may bring in alleles that do not exist in our focal population. Mutation, of course, is the process by which new variation is going to be generated. And recombination is reshuffling of variation that exists in population, but in new combinations through sexual reproduction, for example, or even in species that don't usually sexually reproduce, you might get recombination. And that will introduce some variation as well, okay? Natural selection and drift are two major evolutionary forces that will then act on this variation that exists and will sort it. Some of the variants may be thrown out of the population because they reduce fitness. Under natural selection, if any variants or alleles are beneficial, they have a chance at increasing in frequency in the population, depending on a lot of different parameters. And the whole goal of the next few lectures in this school is going to be to help you understand when is this going to actually work. Our intuition is that if some mutation is beneficial, it should increase in populations, right? And you'll see examples where that doesn't happen. Drift is one reason why that may not happen. So genetic drift is the idea of how, due to probability that operates in small populations, right? The chances that an allele which is beneficial will actually make it to high frequencies reduce because of genetic drift, right? So in a small population, the chance, just by chance, a beneficial allele can be lost. So we'll talk more about that later. But these are the two main reasons why variants will or will not change in frequency in the way you might intuitively expect to see in populations. What kinds of selection do we know of? There is, broadly speaking, positive selection. As the name suggests, it means that a variant is going to be favored over time in a population. It means it is likely beneficial. Negative selection means that a variant is deleterious. It will be generally reduced over time in a population. Purifying selection is an extreme case where any mutations at a particular site are going to be negatively selected. So when we say purifying selection, it means that only that one mutation that exists, or that's, base that exists at that particular position is going to be retained in the population. Any change from that is going to be weeded out, okay? You may have selection, what's called balancing selection of various sorts, which might retain polymorphism or variation in populations. So everything I've said so far, I've focused on saying, okay, there's one good allele, one bad allele kind of situation. What happens to that? There are many cases where people observe actually that in natural populations, there's a lot of variation existing, right? So take any site in the human genome, you might find that if you survey across different populations, even in this room, you might find there are various bases present at that one site, okay, across all the individuals here. The options are only 4, so it's not that there's so much variation. But if you're defining alleles in more and more complex ways, for example, you're saying, okay, what is a string of bases here? How big is this gene? How small is this gene? And so on and so forth. The possibilities are quite large, right? So you may have many different alleles surviving or coexisting in a population at a time. This is something that has puzzled evolutionary biologists for a long time. And we have now a lot of work showing various kinds of selections and interesting phenomena which can retain this polymorphism in populations. So it's called balancing selection broadly. You can have frequency dependent selection. What this means is, depending on the frequency of an allele, it is going to increase or decrease. It is beneficial or not beneficial in the population, right? And I think we'll go through some examples later, but if not, this is pretty standard textbook material. You can find it easily. Heterozygote advantage is another form of balancing selection where, for example, if you have a 1 locus 2 allele model in your head, so big A or little a, that's the classic stuff you're all probably familiar with, right? So if heterozygote has higher fitness, and we'll define some of these terms in more detail compared to these guys, right? The homozygotes, then balancing selection can retain both alleles in the population because heterozygotes are favored, which means that anytime heterozygotes are generated, you're going to also end up generating homozygotes through normal shuffling of alleles in the gametes. All right, third point is spatial or temporal heterogeneity. So if you have, imagine a population exists in small pockets, right? Or there is change in selection pressure over time. And these different subpopulations that are interacting via gene flow to some extent have different selection pressures that are being imposed on the subpopulation that can also retain genetic variation. Imagine a situation where here's ICTS campus, here's NCBS campus, and some butterfly species is able to sort of go back and forth across both campuses, but the big A allele is favored in one campus, the little a allele is favored in the other place. ICTS is more dry, NCBS is a little bit more wet environmentally. That can also retain more allelic diversity in general in this entire population, right? So those are examples of what has. Oh, come on, what? He's coming. Yeah. Or somebody's coming. Okay, any questions about this so far? Yeah. So purifying selection is really negative selection as well. Purifying, we kind of use them in slightly different contexts. So purifying, sorry? Correct, but her question is, how is that different from negative selection, right? So you're right, that negative selection is what is acting on these other alleles, which are going to be purged out. But we say that this site is under purifying selection, whereas negative and positive selection we'll usually use to refer to the alleles that are under selection. So we say that an allele is under negative selection, but that site is under purifying selection. Yeah, same idea system. Any other questions or thoughts? Yeah. Speak up. Where's the mic?
If in a population there are many morphovariants, so it will account for balancing selection.
What are morphovariants?
Like individuals having different phenotypes.
Will they count
and they are
as?
existing.
Uh-huh.
So it is kind of balancing selection.
No, you'd have to do more work to show that or know whether it is balancing selection. That's retaining these different phenotypes in the population, right?
Yeah,
So
and
we'll come to that a little bit later, not specifically morphological variation, but phenotypic variation a bit, yeah.
And ma'am, like you talked about heterozygote advantages, like so sickle cell anemia will be example.
Yes, that's an example of heterozygote advantage, where heterozygote in places where there is high incidence of sickle cell, of malaria, the allele that causes sickle cell anemia is advantageous in a heterozygote state because these individuals don't suffer from sickle cell anemia so much, but they are resistant to malaria, right? So that's an example where the heterozygote has a higher fitness and both homozygotes have lower fitness. One of them because it allows people to have anemia and they have serious trouble because of that, and the other one because it allows them to be more resistant to malaria. Okay. Any other questions so far? Yes.
Is purifying selection is for the gene, like in the context of the, like, saying it is
Yeah, you can also say that overall, perhaps a genome is evolving under purifying selection. You could say that. Is that answering your question? The region of the genome. It's like how you would, sorry, I put a purifying selection over there. Okay. So so purifying selection effectively means that a bunch of other alleles, okay, in that which are not observed at the moment are going to be under negative selection. Okay. At the same time, that specific base that exists at that position is evolving under positive selection. It's retained in the population, right? And so the phenomenon is called purifying selection, that this region of the genome is evolving under purifying selection. Whereas when we say something is under negative or positive selection, we're usually saying that particular mutation, that particular allele is experiencing negative or positive selection. Okay. So it's just from what perspective are we going to describe? Okay. Phenomenon is the same. Does that help? So whether you say that purifying selection is acting at the genome level, at the gene level, at a site level, that's depending on how you're defining it, what you're doing exactly. The concept doesn't change. Okay. In general, I would hesitate to say an entire genome is evolving under purifying selection. That just sounds very implausible to me, but there's no reason it's not impossible. Make sense? If you find some amazing golden genome that's perfect for this and nothing is changing, then maybe yes, the genome may evolve under purifying selection. Yeah. Any other? Usually you won't. You will usually say height is under stabilizing selection, for example, where a particular height is favored and any deviation from that height is disfavored. We'll come to that a little bit as well. Is there a law that you can't? No. But people just would be confused a bit. That's all. But the concept is the same. Yes. Good. Any other? Yeah. That is the genotype.
So if you look at the homozygote and because like one is against.
Yeah. I mean, heterozygotes may also be selected against in both places. I don't know about that, right? I'm just saying that these alleles are favored particularly in both of these cases. I guess if I was, yeah, let's say this is what is favored in each case. Okay, for a diploid. For a haploid, it might be a single letter that we might use. Yeah, question. You need to use the mic. Can't hear you. Yeah.
In plant breeding experiments, like we select pure lines. So it is some sort of purifying selection. Like Mendel did, he selected only pure lines.
It might have resulted in purifying selection, yes. But the thing is with plant breeding or things like that, you're usually selecting on the phenotype, right?
Yes.
And as I hope all of you are aware of, but we'll discuss a little bit more, the same phenotype can be arrived at by multiple different genotypes, right? So when you're selecting on a phenotype, you don't know what genotype you're selecting on. Does that make sense? Which is why I don't know if then I'm actually imposing strong purifying selection or not when I'm selecting for green peas or yellow peas or whatever. Yep. Any last questions? Sure, yeah. Sorry? Yeah, so one phenotype can be caused by multiple genotypes because there's a lot of, well, many reasons, okay? But the point is that, okay, so let's imagine this way. Let's imagine we take height as a phenotype of interest, okay? There'll be many people in this room who will have similar height, correct? But almost certainly with 100% guarantee, I can tell you that they have different genotypes, any two people who have the same height, correct? And you'll agree with this, right? No. The same phenotype can be achieved via multiple different genotypes. Clear? Not always, right? That's the point. Okay, so there are many layers to this. Let's separate a few of them right now, and then we'll come to more layers later on. So one layer is that when we're defining a phenotype, we're defining one specific trait, usually, right? Such as height in this case. Now my height is whatever it is for various reasons, only some of which are related to my inheritance from my parents, right? What genes I've inherited, what combinations of alleles I've inherited. Okay, so let's say he is the same height as me. Almost certainly he has acquired different sets of genes from his parents and alleles from his parents compared to mine. As far as I know, we're not related, right? And so at the genotypic level, there will be lots of differences, right? Now, most of them have nothing to do with height at all, let's imagine. Like a chunk of my genome will have no impact on my height whatsoever. So that's one layer. That if you're defining a genotype, unless you're defining only, let's say, 5 genes which are involved in height determination, you are going to have a lot of differences at the genotypic level. Okay, so that's a question of semantics a little bit. The second layer and reasoning is that phenotypes are impacted both by the genotype as well as the environment, right? So depending on, let's imagine I had a twin and we were separated at birth, right, or whatever. And one of them grew up in a household where they were fed a lot, like way more than I was fed. Maybe they would have grown taller, right? Or they played some sport particularly, or whatever it is, their life circumstance was different. Then despite having very similar genotypes, my twin and I might end up having different heights. Okay, so there are many such phenotypes which are not influenced only by the genotype. The environment has a big role to play. And that's true for many phenotypes. Yeah, so there isn't a direct one-to-one connection between a genotype and a phenotype. A lot of the cases that you've seen in your textbooks are used in textbooks because they're like nice, simple cases to start thinking about genetics and about phenotypes. Okay, like this sickle cell anemia business, right? We know that heterozygotes have higher fitness in that case because of, we know what's, and that makes sense at this point because at the protein level, we know what's changing in the hemoglobin, blah, 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 because of these mutations, right? But that's not the case always. Does that help? Yeah. So there are 2 reasons. There are other more complicated reasons that we might get into later. All right. Yes.
So can we do experiments in lab where we'll take 2 sets of flies or plants or whatever, and give them similar environmental conditions, same food, everything is constant, and some selection pressure. Can we say that even in those cases there can be differences in a genotypic level? It is possible, right?
At the genotypic level or at the phenotypic level?
At genotypic level. I mean, same phenotype but different genotypes.
Yeah, you can have.
So in those cases, what? I mean, just
You're
different.
saying environment was constant, then
Constant.
what is different?
Even then, it is possible.
You're
Right?
saying you had two clones, fly clones,
Hmm.
and then they were put in the same environment, and you're saying their phenotypes should exactly match, and they don't. You're saying.
No, phenotypes are same. Like I'm just taking this example. I'm saying that environment is same. Even in those cases, it is possible that
Yes.
same phenotype, but just different combinations of protein and different biochemicals.
Of course. Yes. Yeah. So that's the further thing that I didn't talk about much, right? That even with a set of genes that are involved in determining a phenotype, there can be other genes that might play a role, which you didn't necessarily expect to play a role on that phenotype, right? So exactly what set of genes and alleles determines a phenotype is a very difficult thing to pinpoint in many, many cases, right? So again, classical Mendelian traits that we have learned about in our textbooks from school are not great examples of how probably most phenotypes actually work. Okay. Sort of, maybe. We'll come back to this a bit later. Okay. Okay, so maybe I'm also realizing that it's worth defining what is phenotype and what is genotype, just so that everybody's clear about this as well. So these are your genotypes, right? And again, a genotype can be defined in pretty much whatever way you want, right? So I can define my genotype based on a single locus here. One locus is one sort of position in the genome, right? What I mean by when I'm writing big A and little a is for a diploid organism such as humans, at any one position along your genome, this one, there's going to be two copies of it, right? One that came from your dad, one came from your mother. And at that same site, what do I mean by sites? We're talking about bases here, right? You might have one copy which has a T, another copy which has an A. Okay, so that means you're a heterozygote for this locus, for this site. Locus is another word which is used in a very elastic way. A locus may be one site, a locus may be a whole gene, a locus may be a whole chunk of a chromosome which has many genes. I should define what I mean when I say locus, but it's an abstract term that's used in a very general way. Okay, so at this locus, at this particular site, I have some heterozygosity. All right. In this case, there are two alleles. Which of these is ancestral, which of these represents a mutation, we don't know. Okay, because all the information we have is I have these two sets of alleles in my body. Now, the genotype can be defined based on a single set of alleles, a single site. I could also define a genotype based on, let's say, all three of these bases. Okay, let's imagine that the exact sequence at all three positions is important to understand some phenotype of interest. Let's say eye color. Okay, so if I want to know my eye color, I might need to know all three bases, and some permutations of these possible three bases next to each other determines eye color. So you might have purple if you have TTT, you might have green if you have AAA, and so on and so forth. So I could also define a genotype in that way, that my genotype is defined based on three sites next to each other, because that's relevant for the phenotype that I'm interested in. Okay, so again, the use of the word genotype and phenotype is quite flexible. You have to define what you're doing every time you use it. Phenotype is anything that we are observing, measuring, etc., about what we can see about an organism. For single cell organisms, people may define phenotype as the rate at which they grow. People may define it as the size of the cell. Okay. For multicellular organisms, like what we see around us usually, you may define phenotype as height. It may be a discrete trait like eye color. It could be a continuous trait like height. Many different ways to define phenotypes. But again, You have to define what you're using so that nobody's confused. Okay, so these are very, very general terms that are used. Now, a given genotype may lead to a given phenotype in some cases. In some cases, here's an example where both AA, big A, and little a, big A can lead to a red flower color, for example. Is the genotype different? Yes. Is the phenotype different? No. Why? Because big A is dominant. You would expect to say here that because the big A allele is dominant, it's producing red pigment. Even if there's heterozygosity, there's still enough red pigment produced that I get a red flower color. Everybody okay with this? These are familiar examples and simple things that it's important to get these clarified in your head. Okay. Any questions about any of this? Yes. Well, for a diploid organism, yes, you think of one pair, right? What about? Yeah, so quantitative traits will often be caused by multiple different loci in the genome. So something like height, for example, in humans, it's clear that apart from environmental factors, there are many, many loci spread across our genome which are involved in determining height. Okay, so here was a one locus model with 2 alleles that we discussed so far. You can have a 3 locus model, okay, where, for example, in your genome and in different chromosomes, let's say there's one site in chromosome 2, which is important, 2 sites in chromosome 1, which are important, and the combination and specific permutation of alleles at these 3 loci determine your height. At each locus, I have the option of having 2 alleles because we are diploid. Okay. Yes. Sorry, what? I'm sorry, I can't hear you. Can you use the microphone, please? Pass it back.
My question is, when we define a genotype, is it always the case that only 2 chromosomes are involved? Like
For a diploid organism, yes, because we have 2 copies. If there's a tetraploid organism,
no, then if the quantitative traits they're located on multiple chromosomes, it can also happen, isn't it?
yes, but one of these set of chromosomes is still part of one copy of the genome, right? This is from mother. And this other copy is from father. Correct?
Yes.
And so even if there are many chromosomes, we still consider them as one genome, right?
Okay.
Maybe that was the confusion. Okay, yes, right? So when we say one genome, we're saying one whole copy of the entire genetic material. And being diploids, we have 2 such copies. If I was a haploid organism, I would have a single copy. Right? Good. Any other clarifications? Yes. Shout.
Definition of phenotype, is it always that in multicellular organisms it is defined at organismal level? Can it be at cellular level? Let's say like my immune cell has some receptor or something.
Yeah, it can be at the cellular level as well. So for example, let's imagine you're measuring something in mice, right? And the phenotype for different mice with different genotypes or environments, whatever it is how many cells of a particular type are they producing? How many T cells are they producing? Absolutely. Or how many cilia are there on their T cells? If cilia are there on T cells, I don't know. But you get my point, right? So again, you can define a phenotype. A phenotype is anything that you can really measure that for some reason you are interested in measuring, right? And that says something about that organism and its function. Usually you won't be going around willy-nilly defining phenotypes just because, right? Usually you'll define a phenotype because you care about that phenotype. It has some functional value. It means something for that organism, either with respect to fitness or function, reproduction, survival, something. Yeah? Yes. What results in dominance? Multiple things can result in dominance. The simplest example I'm going to give is with this pigment production, for instance. So if big A allele is encoding for the for an active enzyme, okay, which makes some precursor molecule into a pigment, whereas the little a allele means the absence of that. Okay, so for example, I'm going from T to A on this chromosome. Going from T to A means that that allele is no longer actively able to produce an enzyme. Yeah. Simple case, many complex, many other cases. All right, shall we move on? Back to slide 1. Okay, so this is where this is how we usually define selection acting at the phenotypic level. It can be stabilizing, it can be directional, or it can be disruptive. If you're making a frequency, a histogram of people's heights, for example, in this room, we might expect to get a Gaussian type looking distribution, right? Where there's some mean height, I don't know, let's say it's 5.2 feet. That's our average height. There'll be some people above, some people below, etc. Right? If we're saying that height is under stabilizing selection, does anybody want to say what we expect to see to this height over multiple generations? Good. So stabilizing selection means that the mean represents the highest fitness category, okay, for whatever reason. And so over time, you'd expect that this mean should be maintained in that population. What does it mean to have directional selection? So as the name suggests, stabilizing is where something is stable, okay, it's going to be stably maintained. What about directional selection? Yeah. So as the name suggests, under directional selection, you expect the mean to shift in one direction over time. Okay, it might be either increasing height or reducing height in this example. Finally, you have disruptive selection, where the extremes are favored more, have higher fitness, in which case, what do you expect to see? This distribution will start looking something like this over time, right? Because these guys have the lowest fitness. Okay. All right. So this is usually how we will define what's happening at the population level, at the organism level, not at the level of genes and genomes and so on.
A particular trait showing different types of selection in different population.
Yes. So the question is, can a particular trait experience different kinds of selection in different kinds of populations? Absolutely. Right? Because it depends on the environment. It might depend on the genetic makeup of the population itself and so on, right? So for example, in some populations, you might have frequency dependent selection that's operating. So in a given population, if some particular allele is at high frequency, it might face a different selection. Different population, the same allele is at low frequency, it might experience different kind of selection, right? Okay, any other problems so far? Any confusion at all? All right. Okay, so what about convergent versus divergent evolution? As the name suggests, convergent evolution indicates independent instances far away in the phylogenetic tree of life, for example, where the same phenotype or the same response to selection has evolved, okay? Again, this is a very abstract, very broad term. You will need to define exactly what you mean. In this case of flower color, we might say that red flowers have evolved multiple times in the phylogeny, okay? And we might, if we have proper evidence, we might be able to say that red flower represents a convergently evolved trait, okay? What kind of proof do I need for this? For example, I have a phylogeny here, okay? And let's say we know somehow that this ancestral flower color was white. This note, is everybody comfortable with phylogenies, by the way? Broadly, vaguely, you'll get into more depth later on today, but yeah, so we're going this is time coming forwards. This is some hypothetical ancestral. Each node here represents what we hypothesize was some ancestor that existed. Okay, we don't actually have these in hand. What we have in hand is these guys. These are the extant taxa or populations. Okay, so there was, we know for some reason, somehow fossils, whatever else, that this ancestor was white. Okay, and we see that so red color is seen in 3 of these taxa that we see right now. Okay, just intuitively, what you might think is that because these guys share a common ancestor, maybe the red change happened here and happened somewhere on this branch. Correct? That's the most parsimonious, simplest way of thinking how this may have evolved. What might be able to say, if we have good evidence for this, we would then say that red color is a convergent trait that has evolved multiple times in the phylogeny. Twice it has evolved. Now, what would be a case where it's not convergent? In this case, these are 2 sister groups in the phylogeny. Both of them are red. It's possible, of course, that independently there was red evolving here, but a little bit less likely, right? This, I would have a hard time saying this is convergent evolution. Most likely, they are both red because they share a common ancestor who was already red. Okay, so this would not be a case of convergent evolution. Divergent evolution would be this kind of scenario where one of the lineages has stayed white, one of them has diverged. Okay, you might also have a situation where this one is now yellow. So starting from a white ancestor, these traits have diverged in these 2 sister clades. That would be divergent evolution. Yeah. Any confusion about any of this? Good. Okay. Oh, we already went through alleles and polymorphism. So alleles, the term alleles and mutations are kind of used interchangeably, or mutants are used interchangeably. But people define alleles in slightly different ways sometimes. So you might find that in literature, alleles are defined as particular sets of mutations that give you a phenotype of interest or something like that. Okay, so don't be fazed by that. At the very, very basic, allele is basically something that is one mutational step away from some other allele that you've been defining. Polymorphism is, as the name suggests, where there are multiple morphs. Polymorphism can be used at the genetic level. We can say if in a population at a particular position, I'm finding many different alleles, okay, that would be polymorphism at the genetic or allelic level. It can also be defined at the phenotypic level, where we can say that we are all polymorphic here. Yeah, there's lots of variation in phenotype in this room. You can define specific traits which are going to be your focus and say, okay, this trait is polymorphic. For example, hair texture is polymorphic in humans for sure. Eye color is polymorphic in humans, etc. Plasticity. Plasticity is the idea where, or the phenomenon where a trait or the expression of a trait changes, is not fixed. So this is a classic scenario where your genotype does not explain your phenotype or does not predict your phenotype. Why? For example, during development of an embryo, as the embryo becomes many, many cells over time, etc., different environmental factors may play a role and alter the final phenotype. Okay? Classic case of plasticity. As the name suggests, something is plastic, it is malleable, it changes, even though your starting genotype was identical. Okay, so plasticity is a huge topic, and I'm happy to go into it a little bit later, perhaps, if people are interested. Okay, so in evolution, in population genetics specifically, we talk about change in allele frequency over time as our critical metric of when evolution has happened or not. Okay, and this is of major interest. You will hear the term fixation used a lot. When we say an allele is fixed in a population, what we're saying is over time, if the frequency of allele A, when there were 2 options, big A, little a, If that reaches 100%, we are saying this allele is fixed in this population, okay? On the other hand, starting from whatever frequency, if it was lost, well, it's a loss, all right? Any questions about this? These, of course, are not the only two fates of alleles. Under what conditions can we get an allele at intermediate frequency? We just talked about this earlier. Under stabilizing, something like heterozygote advantage, right? Balancing selection can maintain allele frequencies at intermediate frequencies, okay? All right. We want to define fitness. Fitness can be defined either in absolute terms, how many offspring is that individual going to produce, or it could be defined in terms of relative fitness. How many offspring is that individual going to produce relative to the most fit individual in that population or the least fit individual in that population, whatever you want, okay? So again, this is a term that's used a lot. Intuitively, once you understand it, you will get it, what is fitness. But it needs to be defined because it can be used in many different ways and you have to be explicit how you define it. So in this example, so fitness is usually termed W. And you'll define it in this case with a one locus, two allele situation. You will define fitness for each genotype, okay? And usually in evolutionary biology, we'll use relative fitness as our main fitness term of interest, because in some sense, as long as you're not completely dead, your absolute fitness doesn't matter, okay, for evolutionary change. Your relative fitness matters. Why? Because the fate of the alleles and your genes, etc., depends on how are you doing respect to everybody else, okay? If everybody else is passing the IIT JEE exam, you're in deep trouble, okay? As long as you're doing better than most others, you'll get in, if that's your aim in life. Yeah, question?
Is fitness always looked at in terms of the number of individuals that like any organisms produce in the next generation?
No, not always. You can define fitness depending on your ideas of what is important for an organism. But ideally, if you think about it, what is most important for future generations and allele frequencies over time? It's reproduction in some shape or form, right? So it's basically how many copies of whatever allele is there, that is the focus of our investigation. How many copies of that allele are going to be there in future generations? That's kind of what we care about in some sense, right? So in the end, you ideally want fitness to say something about reproduction. But of course, if you don't survive, then what good is reproduction, right? So survival, depending on the model system, depending on, you know, the environment, the situation, survival may be a very, very important metric of fitness, okay? Or it may be reproduction, maybe a combination of both, which is so many different ways. Many times measuring survival or reproduction or estimating it is hard. And so people have come up with proxies for that. For example, how fast do you grow? How big are you, right? These are all proxies in different sets of organisms. For example, in birds, in many insects, bigger females are usually more fecund. They will produce more offspring. Given that there's that correlation that people have measured over time, maybe if you're not able to measure fecundity or offspring production for females of a particular bird, you might be able to use body size as a reasonable proxy, right? So there are many times people will use proxies of different sorts. But again, one has to define, one has to be very clear what one is doing. Okay, any questions? Okay, so in this cartoon example, with our two allele situation, we can define, let's imagine this, we are going to define as our reference genotype, okay? And then we can define the fitness of these other two genotypes based with reference to this big A homozygote, okay? So if I set the fitness of this guy to 1, I can now say if this fitness is 0.9 and this is 0.5, what does that mean? These are both deleterious alleles, deleterious genotypes with respect to our reference, right? The heterozygote has a disadvantage of about 10%, okay? And the recessive homozygote has a disadvantage of 50%. Clear? Why do I use this as my reference? In some cases, I might say this is my reference because it has the highest fitness. In this case, it happens to be true. In other cases, I might have some reason to think this is my wild type. Okay, this is what is commonly found, or this is my reference fly, whatever it is. And with respect to that wild type, I'm interested in asking what is the impact of having a little a allele. And so it makes sense for me to define this as my reference. Can I define something else as my reference? Absolutely, up to you. This is just the standard sort of way that people often do it, okay? Again, define it and you'll be fine. Okay, so then in this example, the selection coefficient here is the difference between these relative fitness values, okay? So the selection coefficient, if in a single allele case, in a haploid case, if I have a big A fitness of 1, little a fitness of 0.9, my selection coefficient is 0.1. And that's exactly what I said earlier there, as a 10% reduction in fitness, right? So selection coefficient is telling you or giving you an idea of by how much is a particular genotype or allele better or worse compared to the reference, okay? All right. So what is the source? I forgot, what time is the lecture supposed to end? Huh? Oh. We can continue. Oh, there's a lot. We can continue later.